what is canola? Canola is actually the classification of the oil. However, many people use the term for the plant Brassica species, typically Brassica napus, they produce the state which contain less than 2% russic acid in the oil and less than 30 micromole per gram of glucosinolate in meal. So just in order to know about the background of canola, we can see this triangle which shows the evolution and genetic relationship between 6 species of genus Brassica. Here in this figure, we can see chromosome in red color, blue color, and green color. Chromosome from Brassica nigra and oleracea, they hybridize to produce Brassica carinata. While the chromosome from Brassica nigra and rapa, they hybridize to produce Brassica gentiana. Similarly, chromosome from Brassica oleracea and rapa, they hybridize to produce Brassica napus. This Brassica napus is an important source of canola. While currently in Australia, Brassica gentiana is also produced as a source of canola. For the processing of canola plants are produced for canola seeds. These canola seeds are used to extract 40% of the oil, where nearly 60% of the meal is left behind. This oil is used in margarine, salad, and as a source of canola oil for the cooking purpose. It is also used as a source of biodiesel, while this meal is used as a feed for chicken and cattle. Despite processing of canola for oil, the exploitation of canola meal is very limited. So my research will mainly focus for canola meal. So in order to see about this table shows the composition of the canola meal, where we can see that protein is the major component in canola meal. Here we can also see in this table there is tannin, synapine, phytic acid, and glucosinolate. They are actually anti-nutritional component in canola meal. So these anti-nutritional components are an obstacle to use them as feed for animal. Therefore, we should find out ways to utilize this canola meal. So one of the way is to find out bioactive compound. So bioactive compounds are natural component of food that influences cellular and physiological activities of cell with potential health benefit. These bioactive compounds have been classified into proteinaceous and non-proteinaceous components. So there are different types of bioactive compounds, but some of them are phenolic, protein, peptide, terpenoids, steroids, lectin, and saponins. My research will mainly focus for phenolic, protein, and peptide. So what could be the potential health benefit of bioactive compounds? They are antithrombotic, anticarcinogenic, cardioprotective, immunomodulatory, and antimicrobial. So next, what are these phenolic compounds? Phenolic compounds are actually the chemical compound having aromatic ring with one or more hydroxyl group. They have been classified into phenolic acid, phenolic alcohol, stilbenes, lignanes, and flavonoids. They are present in different food sources, but some of them are like phenolic acid are present in fruit and vegetables, phenolic alcohol are present in olives, grape, and wine, stilbenes are present in grapes and peanut, and lignanes are present in flaxseed, and flavonoids are present in soy product. Flavonoids, they are of growing interest owing to their supposed functional properties for human health, and they have been reported for their anti-mutagenic and antimicrobial properties. So these protein and peptide, they have been classified according to their bioactivities into antihypertensive and encapsulins, immunomodulate, and antimicrobials. They are also present in different food sources, but some of them are such as antihypertensive peptide are present in corn, fish, encapsulins are present in milk and wheat, Immunomodulates are present in fish and soy products, and antimicrobial are present in egg, wheat, and barley. So what are these bioactive peptides? They are the small fragment of protein that may have health benefit. Example is for protease inhibitor, which are the small regulatory protein that inhibits the activity of another protein such as enzyme. So my research objectives are to identify bioactive compounds in canola meal and to analyze bioactivities of bioactive compound in canola meal. My preliminary work is separation of oil and meal, aqueous crude extract, protease inhibitor activity assays, and purification of protease inhibitor, which is still in progress. Here, in this chart, we can see x-axis and y-axis. Here on the x-axis, there are three samples which shows the protease inhibitor activity in 3 cultivars of canola meal, while on y-axis, it is trypsin inhibition unit per gram of the meal. So here we can observe actually a trypsin inhibitor activity where we can see that BLN347 and carousel 10 and rivet 10, and here it shows that BLN347 has higher protease inhibitor activity as compared to rivet and carousel 10. So this result is further confirmed by this figure. Here in this figure, which is native gel electrophoresis of canola meal crude extract and semi-purified fraction of the extract. Here in lane, this lane 1 is the blank, lane 2 is standard, while in lane 3, which is semi-purified fraction of rivet variety, we can see there are 3 bands here in lane 3. While for lane 4, which is the crude extract of the same cultivar, we are only able to see 1 band for protease inhibitor. Here for lane 5, which is semi-purified fraction of carousel 10, we can see there are 2 bands for protease inhibitor. While for the lane 6, which is crude extract of the same cultivar, we can see there is only 1 protease inhibitor. While for the other variety, which is the semi-purified fraction of my cultivar BLM347, we can see there are 3 protease inhibitor, 3 bands of protease inhibitor. While in lane 8, which is the crude extract of BLM347, we are hardly able to see any band. So from this figure, we can see that yes, the protease inhibitor are present in all the 3 cultivar of canola, and further we can also say that they are more prominent in semi-purified fraction of canola extract as compared to the crude extract. So from these results, it is concluded that protease inhibitors are present in canola meal, and they will be purified to homogeneity and characterized. Canola meal extract will be subjected to different assays for their bioactivities. And I would like to acknowledge Faculty of Science Research Higher Degree Scholarship at CSU, Graham Center, and especially to my supervisory team, Dr. Chris Blanchard and Dr. Taur Rahman and Dr. David Luckett. I can see 2 of them. I do not know what Chris is. So thank you. I will appreciate to answer if there's any question.
Thank you very much, Sarah. Are there any questions?
For what reason are you targeting trypsin?
Because it's a standard method to find out some protease inhibitor activity. So it was available, so I used this one.
Is there some sort of activity in the body that you're trying to inhibit, some disease that's associated
Actually, I'm trying to find out some protease inhibitor if they are present in canola meal, because there are some, as I have got one slide with some bioactivities. So if there's some disease like and there is some anti-hypertensive activity, and we can reduce that activity with by using that protease inhibitor. I'm trying to find out that way, but it's my initial stage. So from my literature, I came to know that most of the people are using trypsin inhibitor. That's why I'm trying to use this trypsin.
Follow on that. So trypsin is an enzyme of the gut. If you inhibit it, then you're not going to be able to absorb protein very well. So that, and if you have, say, for instance, a hypertensive, and you have that's an enzyme inside the body, then any bioactive protein that you have in the gut is going to be broken down into its individual amino acids and taken into the blood as individual amino acids. So it won't be bioactive anymore once it enters the body.
So actually, I'm trying to find out some ways that it's not exactly that I will. This is only the trypsin inhibitor. Maybe there are some other inhibitors too. But I just initially I identified, yes, there are some protease inhibitor. Yeah, there's also trypsin inhibitor. Maybe there are some more, and maybe I will characterize them and I will see that how I can work out further for them.
For what reason were you performing native gel electrophoresis to examine your purifications as opposed to SBS page electrophoresis?
Pardon, can you repeat?
For what reason did you choose to use native gel electrophoresis as opposed to standard technique SBS page?
Because initially, if I'm now at the initial stage, I'm trying to purify. Yeah, I do have some results of purification with ammonium sulfate. Yeah, if I came to know, it's confirmed that yeah, they're protease inhibitor. Yes, I will do. I will do STS later on. Because it was just to confirm, yeah, whether they are protease inhibitor. Initial stages to run the gel on for native, and then you can go for STS and whatever other. Yeah.
Other questions? So I've just got a question for my investigation in antioxidants. You mentioned flavonoids and you mentioned polyphenols. You don't look at bioactivity at antioxidants.
Yeah, I do have planned to look at these things, but if the time permits, yeah, because I'm also looking at protein peptide stuff, and I know maybe it's taking too long time, and yeah, I have planned my research to look at protein peptide and phenolics, but yeah, if the time permits, phenolics is my schedule.
Thank you very much. No other questions? Thank you very much indeed.